Dear students, welcome to Odisha Net Academy. Now I am going to discuss about technique 1. Under that, we will discuss the techniques which are required for protein-protein interaction. Okay, density gradient centrifugation. This is one of the technique. You can, with the help of that technique, you can detect whether two proteins are interacting or not. PAGE, polyacrylamide gel electrophoresis. Then size exclusion gel chromatography. Okay, with the help of that one you can detect whether two proteins are interacting or not. Then immunoprecipitation methods, then fluorescence resonance energy transfer method, and YTH, each two hybrid system. So we'll discuss one by one. Okay, so first we'll discuss the principle of all this technique, followed by one of the application, that is protein-protein interaction. How can you detect whether two proteins are interacting or not with the help of this technique? In addition to that, the techniques 1 contains protein DNA interaction also. It includes the method like EMSA, DNA footprinting, and chip method, chromatin immunoprecipitation method. And we also discuss about in this technique 1 IEF, isoelectric focusing, and 2D electrophoresis. So first come to density gradient centrifugation. What is the principle behind that, and how can you detect whether these two proteins are interacting or not? Okay, so this is the separating method through in which the molecules, whether it is protein or any molecules, that can be separated on the basis of their density. So you have to first prepare one gradient, density gradient. Okay, so gradient means difference in density, same solution but difference in density. For example, for your protein, okay, normally for protein, so we are using sucrose solution, sucrose density gradient, and for nucleic acid we are using cesium chloride density gradient. Okay, so as we are discussing about protein-protein interaction, so you have to prepare the cesium chloride solution, your sucrose solution gradient. Okay, cesium chloride only for nucleic acid. One thing is, when you have prepared the density gradient, okay, so lower dense solution remain at the bottom, and higher density remain at the bottom and lower remains at the top. So one question comes, when it is centrifuge, okay, when you are applying centrifugal force on it, then there is a chance of intermixing of all these things, because all these are having same composition, aqueous solution of what? Sucrose. So there is a chance of intermixing? No. Okay, remember one thing, even if their compositions are same, but as they are different, as they are having different density, they won't intermix. The best example see here. Okay, so Indian Ocean and Pacific Ocean both are having aqueous, that is salt and water, but their salt concentrations are different. That's why they are forming a border line like this. They won't intermix. Okay, so that's why there is no questions of intermixing between the different dense solutions. So in this way you can prepare, suppose 50% sucrose solution you can prepare, then put 45%, then 40%, 35%, and so on. So this is the density gradient preparation. Okay, as already I have told you, so for protein separation we are using sucrose solution, and for nucleic acid separation we are using cesium chloride solution. Okay, that is important. Cesium chloride solution important for separation of nucleic acid. Remember that is DNA replication is semi-conservative in nature. Okay, which was provided by or proved by Messelson and Stahl with the help of this cesium chloride solution or gradient, and at that time they had taken N15 and N14. Okay, now come to our main application or question. First question there, how protein-protein interaction can be detected with the help of density gradient centrifugation. Okay, suppose you are provided with protein A Okay, and you are also provided with protein B, and you have to find out whether these two proteins are interacting or not with the help of this density gradient centrifugation. So first you prepare the density gradient sucrose gradient, 7 molar, 6 molar, 5 molar, 4 molar, 3 molar, 2 molar and 1 molar solution. Then for reference, as you don't know what is the density of protein A, you don't know what is the density of protein B, so run only protein A solution, run only protein B solution, and you came to know that protein A solution's density is similar to the 4 molar sucrose solution, and protein B density is similar to the 6 molar sucrose solution. These are reference or control for you. Now come to real experiment. So you keep A and B solution together, that is with what provide them with appropriate incubation time, and after incubation time then run through density gradient centrifugation. So suppose after centrifugation, okay, case 1, after centrifugation you are getting two separated line or band, okay, one at 4 molar, one at 6 molar position. So what will be your conclusion? They are intermixing or they are interacting or not? See, if they will interact, remember A and B interact, then both will join to form one component or one that is a protein complex. So you may get what? One band. So instead of one band, you are getting two bands, and that too similar to reference A, similar to B. So that's why the conclusion is A and B are not interacting. And B are not interacting. Okay, suppose what happens to bending pattern so that your conclusion will be A and B interacting? Now you must get one band. You must get one band. And that to which position? Now you have to take the equivalent density. When the protein of density 4 unit, protein of density 6 unit get intermixed, what is the equivalent density? Now for that you have to take the help of this equation. When volume remains same, at that time the D equivalent, density equivalent is equal to 2 into D1 into D2 divided by D1 plus D2. So it is coming to 4.8. So approximately 5 molar. So you are getting what? The band at 5 molar, means it is interacting. Got the idea? So in this way you can find out whether two proteins are interacting or not interacting with the help of density gradient centrifugation. This is one of the simplest technique. Okay, then we will discuss about higher or advanced technique one by one. Plus in addition to that, you should have some idea about some extra cases, practical cases like this. Okay, suppose you have added same same A protein, B protein after incubation. Then one of the case you are getting band, thick band at interacting position. In previous slide I already have told you the interacting position is 5. So at interacting position you are getting thick band, but at A position you are also getting one band, thin band. So how can you interpret this result? So as you are getting thick band at interacting position, that means definitely A and B are interacting. Then how can you explain this thin band? Now definitely A concentration is more than B. So that's why you are getting excess A. Got the idea? That means A and B are interacting, but A concentration is more. Clear? Then another case. So in this case, so easily you can say here A and B are interacting, but B concentration is more. Now suppose you are getting almost equal thicker band at 3 position, A position, interacting position as well as B position. So how can you interpret this result? Now maybe insufficient incubation time period was provided. That's why before complete interaction running or separation taken place. That's why you are getting free A, free B also, even if they are interacting. So A and B are interacting, but we are providing insufficient incubation time to A and B. Got the idea? Then suppose you are getting 3 bands, A position thicker, B position thicker and AB position thin. That means A and B are not interacting. So these two thick band indicates that these to what these proteins are not interacting. Then how can you explain the middle band? Interacting position one band is there. Now that is for non-specific interaction. The reason, lots of what unknown regions are there. So even if two proteins are not interacting, but due to solvents group, few groups present in solvent that make few protein interact. Okay, similarly the container itself contains some group, through that A and B getting interacting, but the amount of interaction is very less. So these are called non-specific interaction. Got the idea? Okay, so with the help of the simple technique, density gradient centrifugation, you can detect whether two proteins are interacting or not. In the next lecture, I will discuss about other advanced technique to detect protein-protein interaction. Okay, so thank you.